Question number 3 from 2021, Biology End of Course Test. An illustration of how a particular DNA mutation will most likely affect the polypeptide produced is shown. Original DNA strand, mutated DNA strand, and what type of mutation is illustrated. The first of all, we need to know what mutation is. Mutation is any change in the DNA molecule. As you know, DNA molecules are made out of nucleotides, nucleic acids are made out of nucleotides. And as you've seen here in the original DNA strand, we have guanine, thymine, adenine, cytosine. Actually, there's no cytosine here. So basically, they're in order, and then each 3 of those represent one amino acid molecule. And then if there is any change in this DNA strand, it's going to affect our amino acids, and a sequence of amino acids are going to change by the mutation, and then this is going to affect the structure of the protein. As you look at here, you'll see the first amino acid. It's the same here. There's no change. There's no change here. And here, there's no change. There's no change. There is a change here. We need to look at here. See, something happened here. And then our amino acid changed from his to pro. And then here, there's no change. There's no change. And there's no change. So we need to look at this particular amino acid and then nucleotide sequence. On the original one, we have G, T, A amino acids, actually nucleotides. And then something happened, and this codon changed to G, G, A. So the first nucleotide is the same, second is different, and the last one is the same. So there is a mutation at right this point. Here's the mutation. But what kind of mutation is this? So insertion is when you add any nucleotide into the sequence. For example, if it was something like this is our original codon. If it was like G and then something come here and then like say like A, T, A, you see there's an inserted nucleotide here, but we don't see it here. There's no insertion. Translocation is chromosomal mutation. We're not talking about here. We're not talking about the chromosomes here. It's at the DNA level. So translocation is not an answer. Substitution, when one nucleotide is replaced by another one, another nucleotide. Here we had thymine, but it was replaced with guanine. So this is substitution. And deletion is when you had a nucleotide is deleted. For example, if you had like this, or like this, or like this, you can say this is deletion, but it's not happening here. Basically, we have the substitution. So the answer is substitution. We had one nucleotide deleted, not deleted, replaced by another one. So thymine is replaced by another nucleotide, and the whole amino acid changed from one type to another one. Now instead of this, we have pro because of the substitution. It's a kind of mutation when one nucleotide is replaced by another one, and it changed the whole amino acid.